everybody? DtF in the house. Can you hear me, everybody? What is the word? My apologies. I was cleaning out my dab rig. Sorry, you guys. I'm getting the chat pulled up here. How's the audio? Can everybody hear me okay? What's up, Soul Rebel? What's going on, brother? Soul Rebel, George, what's good? How you guys doing? That guy grows, what's up? Let's just shout out everybody real quick. Token Tom, what's up, bro? Anthony, cheers, brother. Coltagen, my brother, hello. Mikey, what's good? Yeah, just let me know. Let me know if you guys can hear me okay and everything. I believe everything should be rocking and rolling. Oh yeah, we're good. We're good. We are freaking balling. Balling on a Tuesday, but it's Sunday. There we go. Oh yeah, dude. Everybody. Super enjoy your Sundays. Relaxing it up before Monday and all that good stuff. So let's pack one up real quick. Got the dab rig all nice and cleaned up just for this. I'm still smoking on that fucking Golden Gauge. I love this shit. Bluesman, what's good, brother? We are just now toking up, my friend. That is what we are doing. That is what is going on. Shout out to Focus V, Carta. That's why I like to smoke my dab rigs, or my dabs with. You already know, we always start the show off with a fat hit, so we are going to get right to that, and then we'll start the show up. A lot of today is going to be some more terminology. We're going to review a little bit of stuff from last week, and then this stuff can be a lot of information, so I'm trying to take it very slowly, just because it really can be information overload. So you're gonna see some common terminology and stuff that we've already reviewed, and then the big thing for today is Punnett squares. We're gonna talk about that and what that is and how that applies to plant breeding and genetics, and specifically what we are doing and what I am doing, which is the Beginners Breeders Project. So yeah, we'll get down to that and talk about it more, but there's a lot more to it than this, but we're gonna keep it pretty basic, still just kind of build on the foundation and get a good understanding of that before we get too in depth, because once again, if you don't truly understand it and you don't know, then you can't really build from there because you're still shaky about your foundation. So we are going to get this worked out and build this up slowly but surely. So yeah, Punnett squares is the main focus today, and just reviews of terms. So yeah, let's get started here. I'm warming up the e-rig right now. Got like probably 10 seconds and then I'm ready to hit this bitch. So cheers. Dabs up. Hell yeah, that guy grows. Best time to do it. I always love just chilling in the grow, doing stuff, having stuff going on in the background. What's everybody smoking on today? Oh shit. It ain't the fucking DTF and Co. Ohio Grow Show if I don't cough at least once, and if I don't hit a dab. So it's tradition. All right, I'm fucking feeling good, son. Oh yeah, Ukrainian cream. Bluesman, yeah, you're good, brother. Eat up, son. Coffee and cannabis all the time. Coffee and kush. Grape Mountain, that sounds fire. Edible kind of day, you already know. Oreos and orange punch terp trees. Nice, Mikey. What's up, Token Tom? I'm doing good, brother. How are you? Just taking the little dab hit before I start up. Every time I go live, and every time, oh, there's my dehumidifier. Every time I go live, I just make sure I freaking have a dab hit ready and get that shit done and get my mind right. Smoke one with everybody, see what everybody's smoking, just check on everyone, make sure their weekend's going good, they had a good week. So yeah, just kind of vibing for a second here before we get into business, and then after that, tea and a toke. Exactly, brother, exactly. You already know what time it is. Doing that a little bit real quick right before we get started, and then I'm gonna keep it short and sweet for you guys, and then we'll keep on building from this shit. I promise you, I'm definitely working on. Hello, what's up, Lori? Oh, making some oil later. Oh shit, son. Trying to keep these short and sweet, building from every previous podcast, and just making sure it's not information overload, because I really do feel like I did that a little bit in the first few episodes. So trying to take it a little bit slower and then really building from there. So yeah, that's what we're gonna do. Hold on, let me get this exhaust fan off quick. Be right back. Gotta go walk the dog. Yeah, you're good, Tom. You're good, brother. Oh, dude, LA Kush and brownies sounds fucking fire. Alright, you guys, let's get started here. Special thanks to Dab Marino, Cultures and Seed Co., P&W Cultivar, and everybody here. And thank you all for your encouraging words and just making it happen, son. Thank you for showing up. Everybody's links and their socials can be found in the bio. Follow them up, sub up, all that good stuff. So today, like I said, we're going to be talking about just more terms and some well, really it's a review of the terms, because all this stuff applies to the Punnett squares. So we'll talk about Punnett squares and who made those and why that applies, and just review terminology, and then we're going to keep building from there, because there's a lot of information. Also, side note, Cultigen, great guy, amazing dude. Got to meet him this weekend, and it was really good, really awesome. You know, it's just one of those things that sometimes when, you know, you meet people and, you know, it's just kind of like a blessing. So you got to make sure you cherish those people, and if you don't, then they exit your life. So you got to make sure you act accordingly, because, you know, I think we have all experienced that type of stuff where, you know, sometimes you just kind of don't act like yourself, or you just mess up, and then you kind of have to deal with the repercussions of your actions, or like you lose a good friend or a good good woman or a good anything. You know, you can ruin anything just because you're not doing the right stuff. So like, when people take the time out of their lives to invest in you and support you, you have to invest in them and support them, and then that in turn just kind of builds builds character in both of you. So that's my little side rant for today. But I met him, and it just was a really good experience, and it was cool. And yeah, man, he's a great dude. And shout out to you, brother. Shout out to George. Shout out to everybody over there, man. It was a great time, man. So I'm a sleepy guy though, dude. Yeah, I be hitting these dabs and be fucking way too tired sometimes, man. I can't stay up late. But yeah, dude, Coltagen, he's a hell of a breeder, man. So shout out to you, brother, for real. Just wanted to say that before we get started here. So basically, like I said, we're gonna review some terminology. But first, we're gonna talk about Reginald Condone. He was born in 1875 and he died in 1967. He studied at Cambridge University, and he developed these Punnett squares. And the reason he developed these Punnett squares, okay, so he talked about Gregor Mendel in the previous presentations. He kind of helped with putting Gregor Mendel and Mendelian inheritance on the radar. So like him and some like-minded individuals, English people, they were introduced to his work, and then they were spreading it around over there. And then he was developing different ideas about it, and he was talking with his colleagues about stuff, and that's essentially how they were figuring stuff out. So a Punnett square is like when you use it correctly, it will allow you to begin to visualize your plant crosses with a simple prediction by using the dominant and recessive alleles as the determining factor of how your results for your Punnett square are shown. And that's just my kind of little summary on it from what I would say about it if somebody asked me about a Punnett square. So that's kind of what I thought. And then now I'm going to give you like a more of an official definition or description of a Punnett square. So a Punnett square is a chart that allows you to easily determine the expected percentage of types in the offspring of two parents. Both parents are heterozygous, which that's another term we're going to review, but that just means it has two different alleles, essentially. So you'll see it here. I'll just say one is a big P and one is little p. Just think about it like that. That's what heterozygous means. It means there's two different ones. One's big and one's small. Just think about it like that. But we're going to review that here along with the Punnett squares, but just hold on. So the different possible combination of alleles in the offspring are determined by when you fill out this Punnett square. So you basically will use these heterozygous big P, little p, and then you will do a cross to that. So it would be another big P, little p, and that's what you call a monohybrid cross. And then what you do is then now you make a Punnett square, but I'll show you an actual example of that coming up. So the different possible combination of alleles in the offspring are determined by filling in the cells of the Punnett square with the correct letters, or the letters represent the alleles. So basically that's just telling you that it's all tied together. So like I said, it can be confusing sometimes. So let's just get into the meat and bones. So Hardy-Weinberg law is another really important thing too, but that's a completely different area, and we're going to get into that more too. But basically what that means is, and what it is, and how it came about is, like I said, Punnett was talking to his colleagues, and one of his colleagues happened to be G.H. Hardy. And Hardy was a statistical guy, and Punnett was coming from his side of genetics, and he was basically asking Hardy, so if they know that brown eyes is a dominant trait in humans, and they wanted to know, well dang, if it's a dominant trait, why is there only an X amount of people that have brown eyes, and there's still recessive traits, or people with blue eyes or green eyes, or that sort of thing. So that's kind of how that's tied together, but that's something else to be discussed. So basically we're going to review these terms, and then I'll show you the Punnett square stuff. So genes. Once again, this is all going to be stuff you've seen before. Besides the Punnett square stuff, I just want to re-emphasize what this stuff is and how it plays together and it intertwines. So all this stuff is a part of Mendel's, and it's building from Mendel's inheritance and Mendel's 3 laws. And all this terminology, it all is applying to the field of genetics. So, and I just want to emphasize that it's important to take note of what these things mean, because they're going to consistently keep appearing and constantly keep appearing. And it's just good to know what it means. And once again, if we don't have a solid foundation, we can't move forward to keep building. You can't build a house with a shaky foundation. It's going to fall over. So just think about it like that. It's the same concept as that. Your learning is just like that. You have to start out with a good foundation and build from there. So that's why some of this might sound redundant, but just want to emphasize that it's important to know. So genes. What are genes? Genes are parts of DNA, and they carry the hereditary information passed from parents to children. So genes contain the blueprint for each individual for her or his specific traits. So if your mom has brown eyes and your dad had blue eyes, the dominant trait is brown eyes, so more than likely you're going to end up with brown eyes. But you could, because of recessive traits, end up with blue eyes. You never know. So it just depends on if your grandparents, what they had. So it's all tied together. So that's just kind of what that means. So next one, allele. We already talked about this too. So allele is a different form of a gene which produce variations in a genetically inherited trait. So this just means that it's a different type of trait that you would get from your parents. So different alleles produce different hair colors. So that's exactly what that means. If you have red hair and your dad had red hair, you probably got that gene from him. And that's basically what this is saying. So a dominant allele, just like we were just talking about, dominant and recessive, a dominant allele would be just like the brown eyes being dominant over a blue eyes. So dominant versions of a gene shows it's a specific trait even if only one parent passed the gene to the child. So when a child inherits the dominant brown hair gene, the child will have brown hair. But once again, there's recessive traits. And recessive traits will carry on, but be passive as they're carried on. Then once there's a combination of 2 recessive traits, then that's when that appears in the phenotype. So the gene or the genotype is that gene that makes you physically see somebody with brown hair. So the gene is what physically is being expressed when you are seeing it in real time. If I would meet one of you in person and you saw me, I have brown eyes. So that's my genetic genotype being expressed through my physical attributes. It's my phenotype. I genetically got the genes from my mom and dad to have brown eyes, and that was passed down to me, and now it's physically being expressed through me via genetics, because that's the way it is explained and the way that Mendel worked it out to be. And then once again, we're still building from that. So yeah, recessive allele. Recessive, like we just said, it passes on, and when 2 recessive alleles combine, that's when you then see the recessive trait. So if you had your mom or dad that had brown eyes and one had blue eyes, and you have brown eyes, and then you have kids and then your kid has blue eyes, it's probably because there's a recessive trait being passed down that is now being expressed through your child, through the genetic gene pool that's being passed down and inherited through each generation, which once again we're tying it all together. So when a child inherits recessive blue eye gene from both the mom and dad, the child will have blue eyes. That's basically what it's saying. But then that kid necessarily won't have blue-eyed children. That's just that, and that's how it works. It's the genetics playing a role in that. So heterozygous. So this is what I was talking about with the 2 letters being represented, one big and one little. So that would be heterozygous. If it was the same, if it was 2 big letters, it would be homozygous. So heterozygous is 2 different forms of a gene. one from your mom and one from and the other from the dad, and they're both different. If you inherit a different eye color, from your mom and dad, then it's going to be expressed when it combines, when those two alleles combine and the gene is formed. That is when it is decided, okay, this is now going to be a brown-eyed child or a blue-eyed child. And then that's the random part of it, or Mendel's third law of independent assortment. Yeah, so there's we can't forget about Mendel and the law of dominance, the law of segregation, and the law of independent assortment. So that's what that's talking about. When there's two different forms of the gene, they'll randomly separate, and then when they come back together to combine, that's when the law of independent assortment comes into play, and then that's when it is decided whether you're going to be blue-eyed or brown-eyed. So that's just what this is saying, essentially. So homozygous, like we just said, it'd be like having two big letters. So if it was big P and big P, that'd be homozygous. Or if it was big P and little p, that'd be heterozygous. And it's just two of the same form of the gene, one from the mom and one from the dad. So if both your parents have blue eyes, you're probably going to have blue eyes too. So once again, we're talking about Mendel because we're building from that too. So Mendelian inheritance is just the simple genetic rule where a gene only comes in a dominant or a recessive form. And as this is being followed, so some genetic traits will follow inheritance and some won't. So that's just what that is talking about. And once again, the genotype and the phenotype, that's just talking about P, or oops, my bad guys. P for me equals phenotype. That's how I remember it. P equals physical, and then G equals genotype, because G equals genes. That's just how I narrate it in my head to remember it. So that's that. And then once again, the example I like to use is the genotype and the phenotype of the indica and the sativa plants. So sativa plants, the landrace plants, are known to have narrow leaves with tall, lanky growth. Indica, in comparison, is broad leaves with short, compact growth, and that's just because of the different genes being expressed through the different environments that they're in. So the environment does play a role in what physically will be expressed, because the environment is what's dictating its genotype. The plant is learning over time, and as plants are coming into season and dying each year, the ones that are the strongest and adapt the best are the ones that are surviving. So then that's the ones that pass on their genes. So that's just what this is saying. Genotype, genes that individual carries and passes on. The strongest of the plants will pass on their genes, and then that's what's that's what's going to be physically being expressed mostly, because that's what is surviving the best in that environment. So the sativa leaves have the narrow leaves and the tall, lanky growth because that is what is needed for the equatorial environments, where it's a lot hotter and a lot more humid than typically in the indica environments, which are a lot harsher and a lot colder, a lot lower humidity. So there's different growth patterns that are needed for the plants to survive, and that's why there's different physical attributes to those plants. But it's still the same family. It's still the same plant. And then once again, that's also why people have different colored hair and different colored eyes, and different people are different, like taller and shorter. It's just a part of all that. But we're talking about the plants, but it's all interchangeable when you want to talk about the genotype and the phenotype. So yeah, that's just what that's talking about. So now here we go. We're about to get into the Punnett square stuff. Thank you for sticking with me. Monohybrid and dihybrid. So we talked about this before as well too. So monohybrid is when one trait is considered. So let's just tie it back to Mendel and his pea plants. So he took his tall pea plants and then the short ones, crossed them, and in this first generation, that was the F1 progeny, and they all turned out to be tall. So then, therefore, we can assume that the dominant trait would be tall, and then the recessive trait would be short. So then he took this F1 progeny and he crossed that, and then he's seen that there was tall and short, and that is because there was recessive genes still being passed on, even though all of the pea plants were tall. When they were crossed again and they had another generation, the recessive trait then combined with another recessive trait, and then that made the short plants. So that's why there's tall and short. So it was a 3-to-1 ratio, and he developed multiple generations of offspring, and he kept recording this data. And this is actually something that you will have to do if you are trying to breed, and if you are trying to take note and take different traits and continually pass them on to your offspring. So that is another reason why this ties into the Beginners Breeders Project. You will have to take extensive notes, and Cultigen, he will tell you himself, you have to be very mindful and plan what you're going to do and what's happening. And that is exactly why I am currently growing out some of my one cross that I made of the Chill Mints OG, which is the Chill Out OG by Mr. Grow It, his F3s, crossed to my Runt Mints, which is CRV Mints crossed to White Runts. And so basically I'm growing these out right now just to see what the morphology is, how they grow, what type of stresses they can endure, and I just want to see what happens. I'm not going to train them. I'm not going to treat them any differently through waterings. The only difference between the other plants is that they are going to get trained and these ones won't, just because you have to take note and take heed of what you're even growing. So I mean, yes, you do need to eventually test and see if it can take training in high stress and low stress, and if it yeah, you do have to take note of all that stuff too, but you got to make sure that the plant is worth continually breeding with too. So you don't want to keep growing. If I have let's just say somebody has they're at a I know everybody is in different states and they have different laws and stuff, but let's just say you have 10 seeds or whatever, right, and you pop all 10, right? If you see that some are more drought resistant than others early in life, and then they exhibit that through vegetation, and then they exhibit that through flower, that's something to take note of, and to definitely see if the other plants adhere to that same too. So then if you look back and you're like, well, no, this one actually wasn't drought resistant, because you just have to take notes. That's all I'm saying. So the dihybrid cross is 2 traits considered, each with 2 alleles. So there's dominant and recessive, and then once again tying it back to Mendel, Gregor Mendel, he took a wrinkled green seed pod and he crossed it to a smooth round yellow seed pod, and then he's seen that the first generation, or the F1 progeny, changed the colors of the pea pods to yellow round. So then he stated that the dominant traits are the round shape and the yellow color. He then self-pollinated this first generation of progeny, and then he had 4 different traits: round yellow, round green, wrinkled yellow, and wrinkled green, and the ratio of 9-3 to 3-1. So once again, this is just tying into the Punnett squares, because you can do a monohybrid Punnett square representation, or a dihybrid Punnett square representation. And basically what this means is that you're taking 2 traits that you have observed inside of your genetic pool. So let's just say it's big bud, little bud, or it's green bud, purple bud. So if those are traits being considered, then you can use these, and if you have extensive notes and you know that this is something that is consistently happening, you can take these notes and make Punnett squares to help you theorize and predict what your outcomes will be when you take a big bud to a little bud or a green bud to a purple bud. So that's essentially why this stuff plays in and ties together. And I apologize for sometimes rambling and stuff. I feel like I ramble, so ramble. I think that's a Led Zeppelin song, I believe. Ramble on. Yeah. So, okay, bam, bam, bam. I feel like I need some water. Man. Okay, so monohybrid cross. So, we were just talking about the heterozygous, which is having one big B and one little p, and we're going to call this heterozygous dominant big bud. So, don't let heterozygous confuse you. It just equals different. So if you want to think about it like this, just know that heterozygous means different. Homozygous means the same. So, okay. We're going to have the representation of heterozygous dominant big bud is represented by big B, little b. The homozygous recessive little bud is represented by little b, little b. And then the homozygous dominant big bud is represented by big B, little b. And then also we have heterozygous dominant green bud, which we're going to call GP, and then we're going to have homozygous recessive purple bud, and we're going to call that PP. And then we're going to have homozygous dominant green bud, and we're going to call that GG. So if you then look at this next picture here, the monohybrid cross, would be the heterozygous dominant big bud to another heterozygous dominant big bud. And then what would happen here is you have the big B and the little b represented by this square over here that you see. And I really truthfully apologize about my handwriting. I'm sorry. But the first square right there, if you take a big B and a little b cross to a big B and a little b, the first Punnett square that you are going to see, I don't know if you guys can see that cursor right there, it's going to be crossed to a big B and a little b, or a big B and a big B, and then you will get in that homozygous dominant big bud, because that's representative of the big B, and then you'll have two heterozygous dominant big buds, and then one homozygous recessive little bud. And all this you can see is represented over here in this Punnett square. So when you take two big B's crossed to each other, that equals BB. When you take two little b's crossed to each other, that equals lowercase b. And then you can see that here. So there's going to be a 75% ratio dominant big bud being shown. Damn, sorry guys. And then 25% recessive being shown. So then this is just telling you that when you cross these together, you're mostly gonna see the big bud being crossed down to the first generation of offspring with very minimal little bud. So that's basically what this is saying, and that's the monohybrid cross. And then if you want to look at it from the color perspective, it would be the green bud and the purple bud, and then we have GP, we have PP, and GG. Okay, so the G, the capital G and the lowercase p is the heterozygous dominant green bud. The homozygous recessive purple bud is lowercase p, and the homozygous dominant green bud is big G. So when you take a monohybrid cross of the G, the capital G, lowercase p, and you cross those two together, you then get, just like above, you get two capital G's in the first square, and the next one you get one capital G capital P, and then once again you see capital G, capital P, and then P, P. So do you see the ratio again is 75% dominant trait being passed on, 25% recessive trait being passed on. So that's what you can also look at this as, is that 75% of the time when you take a monohybrid cross, you will get 75% of the dominant trait being passed on, with 25% of the recessive trait being passed on. And then when you take those and cross those again, then you would have another different ratio of plants. So what this is saying is that you can take these, if you have extensive notes of, okay, this one has, let's just talk about it in the form of terpenes. Okay, so this terpene is pinene, and this terpene is terpinolene. I think that's how you say it. And I want to have more of the pinene. So then you would then have to take notes of, okay, well, this one was more leaning towards this or that, and then you can take a representation just like this and make up letters like how you would want. If it's, let's just say for example, it's big P and little t, and then the P is representative of the pinene, and the terpinolene is representative of the little t. So you would have to figure out first which one's dominant and which one's recessive before you even do the Punnett square. So you'd have to do crossing and then determine, okay, it seems to be that this is like you'd even have to really go and get terpene tests and all that stuff done if you really wanted to go that far. So you would actually know for 100% this is what I'm passing on to the next generation. So I can't even sit here and theorize or speculate. Like you honestly would have to go get a test done on your stuff so you can figure out what is being passed on. I guess that's the way I would want to do it officially. I also am not gonna do that though. I'm just doing this stuff for fun for myself, so I'm gonna just try to take really good notes and maybe one day do that. But yeah, so that's just what this is talking about. This is just, once again, a monohybrid cross, because you're taking a big B and a little b and crossing it to another big B and a little b, and then the Punnett square of the big B and the little b cross to each other is represented at the top on the right hand side. And when you do that, you have a 75% ratio of the dominant trait being passed on and 25% ratio of the recessive gene being passed on. And this can be determined because if you look, 3 of the squares have all capital B's. 1 of the squares has a lowercase b. So those 3 capital B's is representative of the 75% of the dominant trait being passed on, and those 2 little b's is representative of the recessive trait being passed on only 25%, because that's the final fourth of the whole 4 squares of the Punnett square. So yeah, 3 out of 4 is big B, 1 out of 4 is little b. Same thing with the one below, with the monohybrid cross of the capital G, lowercase p, cross to each other in the monohybrid cross. You then get 75% or 3 of the capital G's of the dominant trait being passed on, and then 1 or 25% of the recessive gene, little p, being crossed or being passed on. So then now you have a dihybrid cross. So this might be a little bit, my bad, that's my cats. Hold on. Okay. So the dihybrid cross here, it's a lot more complicated because there's more squares, but it can still be done. So the reason that there is more alleles or more combination is because you're now considering 2 traits or 2 genes with 2 alleles. So there's 2 genes with 2 alleles. So 2 times 2 is 4 because there's 2 genes with 2 alleles. So that means there's 4 alleles. 2 genes with 2 alleles. 2 times 2 is 4, so 4 alleles. And then, as you can see, there's 16 different squares or combinations that are made from this. So another reason why I had the representation on this left hand side of all of the combinations of the big B, little b, and so on and so forth is because it's important to take note of this, because then this is what we're now taking. We're taking a big B, little b, big B crossed to a capital G, little p, capital G, capital G. So then when you start crossing these inside the Punnett square, once again, sorry for the handwriting, when you cross these in the Punnett square, you can see that all these combinations are going on here. And this is another reason why you have to take extensive notes if you're going to be doing this stuff. It'll just help you to keep track of what genes you want to pass on. So let's just take note of this again. Just know that if there is a capital B or a capital G, that's the dominant. These damn cats, I tell you. Just know if there's a big B or a capital G, that is the dominant trait being passed on. If there is lowercase anything, that's the recessive gene being passed on. So with the dihybrid cross, once again, there's 16 combinations because there's 16 squares. If you counted these up here, starting from the first one, there's 1, 2, 3, 4. There's 4 in each row, and there's 4 rows, so that's 16 possibilities. And then, like we were just talking about, if you see the recessive and it's all lowercase, then that means that's a recessive combination. If you see one that has and that means also that the recessive trait is going to be shown. If you see one that has the dominant traits only, then that means that the dominant trait is going to be shown. If you see one with mostly dominant traits being shown, then that's going to be mostly dominant but some recessive. So on and so forth. That's basically what this is saying. Once again, thank you for sticking with me. One more time, we'll just look at this real fast here. I just don't want nobody to be confused, and sometimes I get worried about I may not be explaining it well enough. So yeah, once again, if you see any capital letters, that's representative of the dominant trait being passed on. If you see anything with lowercase letters, that's the recessive trait being passed on. And that is represented once again by these crosses over here on the right hand side, but then also the dihybrid crosses too. Just trying to give you guys time to look at this a little bit here. Oh, whoops, sorry. So doubling back and talking about G.H. Hardy, the Hardy-Weinberg law was developed because somebody asked Punnett what and why brown eyes are not the only thing that is being expressed and seen, and he could not describe why that was going on. So then he talked to G.H. Hardy about it, and when he did, he came up with basically a calculation to determine what happens with population and dominant traits. So we're going to talk about that on the next presentation, because that's a lot more information to do and stuff as well, and I feel like we've went pretty far. So yeah, that's what I'm about to be calling it here. We're gonna do another dab hit here and basically be skedaddling, you know. So that's what's gonna be happening here. I hope that you guys had at least some information that you learned, and I appreciate being here with me. I'm about to load up this fucking dab right now. Sorry if it may seem redundant at times on the presentations, but once again, it's very important to emphasize the terminology because it's stuff that's going to be continuously coming up. So yeah, big takeaway from this is that dominant traits will mostly be passed on and expressed physically and seen, but just because there's a dominant trait, that doesn't mean that the recessive trait is gone. It's still passed on passively. When you see a recessive trait being expressed, that is because there's two recessive alleles being combined together, and when those two genes come together, they are now deciding, okay, the only possibility is to express this recessive gene because that's the only two alleles that are in the equation. So when that happens, then you see the recessive allele being expressed. So yeah, and then you can see that being represented in the Punnett square by the big B's and the little p's, or the big B's and the little b's. And that's just telling you that there's a dominant. If it's two big B's, then that's telling you that there's two dominant alleles. If it's a big B and a little b, that means there's a dominant and a recessive allele, but the dominant allele will be expressed first, and then if there's another generation, then and another recessive allele is combined with that other recessive allele, then you'll have a recessive phenotype trait be expressed, and it will be different than the dominant trait. So that's why they say to always take, and Cultigen told me to always take your crosses and your inbred lines to more than one generation, because yes, you can have a F1 generation and you will see a lot of genetic variability and a lot of vigor, a hybrid, they call it hybrid vigor, but if those plants are then taken by somebody else and you didn't test them more than just the first generation, if somebody takes those plants and takes it to another plant and a recessive trait was passed on that was bad from the first parent, and then a recessive gene from the second parent's passed on, and then those two combine, and now it's like completely detrimental to your grow in your garden, then you know you're kind of just shit out of luck. So you have to be aware, and you have to be willing to do the work and the notes to understand, okay, this is what I want to be passed on, this is what I do not want to be passed on, because you have to know what you want just as much as what you do not want. For me personally, I think my biggest capacity or my limiting factor would probably be the fact that I only have a 6 foot maximum for my height. That's as high as I can go. So for me, it's going to be really essential to make sure when I'm breeding these plants and I'm trying to cross my own genetics and genes, that I'm doing it for my environment, for what I'm doing, and that's what I encourage you guys to do as well too. But I'm gonna have to continuously keep growing and breeding the plants, and continuously taking notes to know, okay, mostly it's tall. Oh no, mostly they are drought resistant. Oh no, and then oh no, when the next generation came, they weren't. It was this. So like, you have to take notes. You have to be very, very attentive to detail, and be willing to learn and just keep working with them, because that is how you end up with something that you want to keep. And yes, sometimes you'll get the one offshoot of something that's just really, really awesome and really cool. But sometimes, what if you want to take that thing that was really awesome, really cool in that one plant, and you want to pass it to the next plant? So how do you do that? Then that's how that's why we're doing this. That's why we're talking about this stuff, because we want to take the traits that we want, and we want to pass it on and make good, ethical crosses that are strengthening the genetic gene pool within cannabis and not weakening it. We don't want to pass on nasty, underdeveloped plants, nasty plants that just don't do what you want them to do. If your environment requires you to have plants that are taller or plants that are shorter, you need to take note of that. If your plants are in an environment where they require more or less sunlight, you need to take note of that, and that applies to your crosses too. So it just takes a little bit of work and a little bit of time. And then once again, that's also why I want to keep reiterating the same foundations and the same terms, because it's just one of those things where if you don't understand it, you won't be able to move forward with what here's how I would put it. If you don't understand it, you won't be able to move forward with intention. And Cultigen told me that you have to plan, and you have to be attentive and intentional with what you're doing with your plants and with your crosses. It's not just some guessing game. There is some rhyme, and there is some reason to what you're doing, and how you can make the plants work with you and work for you. So you just have to be willing to work, and have to be willing to take notes, and just keep them freaking growing, and keep being interested in what you're doing. That's the number one thing too. Be interested in what you're doing. If you're not interested in plant breeding, then don't do that. If you are interested in it, then do it. Don't be on the fence about it. Just do it. Oh damn, guys, I'm so sorry. I just burped. My bad. I'm so sorry. I apologize about that. That's probably really loud. Oh shit. I thought it was a yawn, bro. I'm not even gonna lie. Alright, time for a dab hit, and we're out of here. Thank you for coming. Thank you for stopping by. It's Dab 30 all the time. Hey, that rhymes. Next week we will be talking about the Hardy-Weinberg equation and law, and what that is, and what that has to do with Mendel and genetics and everything that we're doing, and why it plays into genetics. And that's what we're going to talk about. And we'll probably add a couple new terms. Cultigen gave me a recommendation of doing codominance and just stuff like that. So yeah, we're going to get some more information cultivated for y'all, and we're going to go from there. But for now, it's time to hit a dab. Cheers. Nothing like smacking a fat dab. Okay, everybody, I think that's all she wrote. Big shout out to everyone that stopped by. Big shout out to Coltagen, to PNW Cultivar, to Dab Marino. Big shout out to everybody, man. Like, I really, really do appreciate you guys stopping by with me and rocking with me. And yeah, we're gonna keep on going. I'm really excited about the re-veg plants coming up. Like I said, I saved some of the Milktopia. I saved the Georgia Pie. Want to get those re-vegged. Take clones. Bada bing, bada boom, you know. So far, once again, like I said, I'm gonna get more content going out here too. So let's just talk about that for a second. New content on the way. Right now, at this point, I'm just waiting for the stuff to cure up so I can do smoke reports on it. And then once I do the smoke reports and I take pictures and videos of everything, I'll do the compilations of them and I'll get some videos out there. But yeah, just waiting to have them be. Basically, I'm waiting to have them be to the point that I'm ready to share the information about them. You know, right now I just feel like they can go a little bit longer. Don't get me wrong, of course I've been sampling it here, and right now my favorite is the Milktopia. If you've been rocking with me this whole time and you've been seeing my Instagram and my YouTube, you know that I was really hyped up about the Milktopia, and that's the number one that I'm really leaning towards right now. It has a really nice smooth hit, but when you also hit it though, it's kind of like this. Man, it's kind of like this kind of pungent taste, but smooth. It's like a smooth and pungent combination is what's going on. But I would really like to eventually, like let's say I end up wanting to take whatever terpenes inside the Milktopia. I would be willing to do a test so I can determine what that terpene is. So then maybe I can try to get that terpene worked in somewhere. But right now, that Milktopia is fucking great. Yeah, probably gonna be another week on cure or so. Maybe not that long. That sounds a little bit excessive actually. But yeah, I mean, that's basically what's going on over here on this side. We're just trying to get these plants re-vegged. I'm not even sure when that's gonna happen. If you guys know how long it takes to re-veg a plant, drop it in the comments of the video. Hit me up on Instagram, tell me. And yeah, I mean, I don't have really much more to tell you guys or to go over. I'm pretty much done with what I was talking about, and I need to eat some supper. I need to get some plant stuff done, and yeah, it's about that time. So I think that's what I'm gonna do, you guys. You already know. Many blessings to you, to yours. Keep on growing. Keep on smoking. Peace and one love. DTF, everybody. Down to funk.